Okay, so good afternoon, everybody, and welcome to Dear Editor, which is our ongoing series in the library about scientific writing and publishing topics. And today we're going to be talking about how to create a graphical abstract. And most of this topic is going to be covered by my colleague, Amy Nanetto. Um, and so I will turn myself on mute here in a moment. For those of you who are tuning in, we will have the slides and a link to the recording, and we're also going to have a handout with additional resources that goes along with today's topic, and I will send all of those out to attendees after the session, and also post them on the Dear Editor team. So with that, I will go ahead and turn it over to Amy.
Thanks, Erica. So some of you may have already made a graphical abstract, some of you may not have. So what I'm going to talk about today is some basic tips for some things you want to think about when you do construct a graphical abstract, when and if you ever have to, some advantages of using them, and some tools you can use to construct them if you're not, like most of us, not a graphic designer or an artist. So the first thing, obviously, is what is a graphical abstract? And I should say I'm going to use the terms graphical abstract and visual abstract interchangeably in this presentation. Journals sometimes use one, sometimes use the other, sometimes mix them up, but they're the same thing. So what are they? So the publisher, the big scientific publisher, Elsevier, says that a graphical abstract is a single, concise pictorial and visual summary of the main findings of an article. BioRender, the graphical design tool for biology, says something similar, a single image that gives your reader the main message of your science story. I'll skip that last one for a second. So you can see these definitions are really similar. They both say it's a single image, focuses on the main findings, main conclusion, or the main take-home message of your story. The Elsevier definition says that it should be concise. So that's a similar thing to the written abstract. And then this last one is from a blogger for Springer Nature, another big publisher, and says that the graphical abstract is kind of like a movie trailer that gives away the plot. So a movie trailer is, designed to generate interest in seeing the whole movie. And sometimes we don't like it when it gives away the plot, because then why would you go see the whole movie? We don't want to see the twist at the end or all the funny jokes. But the graphical abstract is a little bit different in that it should give away the plot. We want to focus on not on the study itself, the whole story, but really on how the story ends, what the main conclusion is. So it's not designed to create suspense. Visual and graphical abstracts have become common, really since journals have moved primarily or solely online. That's the reason that they were adopted, to give kind of an easy visual framework for people to screen articles and to generate interest in them. So one reason you might want to use one is that they really do help improve your article's reach to wider audiences and to interdisciplinary audiences. Most journals that have social media sites and use graphical abstracts post them on their social media sites. So this is from Instagram. They usually also put little thumbnails of the visual abstracts in the online table of contents. Sometimes they get featured on the journal website for a feature article, or even parts of them or the whole thing, sometimes on a cover image on the journal, which is really nice for spreading the word. Institutional websites sometimes use them. In institutional newsletters. I don't see MD Anderson doing this, but some institutions do. You can put them on your website or your lab's website. You can put them along with your paper on preprint servers, on your personal or lab social media sites. So there are a lot of ways that the visual abstract helps get the word out. to a variety of audiences. And there is some empirical evidence that visual abstracts increase engagement either with the abstract of your article or even the full text. So this study from 2017 compared tweets that contained a visual abstract with tweets that just had the titles of the articles. And it's sort of, if you use social media, you understand that, posts that have pictures in them or videos get more engagement than posts that are just text. And that's what they found, that there were substantial increases in the number of views or impressions, the number of retweets, and a smaller increase in the number of visits to the entire article. Now, some other studies have suggested that that effect isn't as large as this one. There have also been studies that suggest that video abstracts really work well to generate visits to the full article. And if you can, summarize your article on TikTok, it's probably going to get a lot of engagement. And, I don't know, perhaps we'll do a future Dear Editor on video abstracts. So I'm going to launch another poll. You see the poll that launched. And this one is, do you look at graphical abstracts? Okay, let's do just like 5 more seconds. And Erica, I don't know, can you see the results? I'm not sure if it's, oh, here we go. Okay, so similar numbers. Have you said that you found them helpful, or have you looked at a few of them? About 10% of you said, I ignore them, and 13% said you actually haven't seen one. So they're certainly not universal. Some journals use them. I would say most don't yet. But I think they're probably becoming more and more useful as journals try to improve their social media presence and their online, and kind of the attractiveness of their online profiles. So in looking through hundreds of graphical abstracts for this presentation, I developed kind of a typology of 2 main styles that are used. One I'm calling the infographic style because it uses kind of the little icons that look like infographics. And it kind of, you could call it the mini poster style as well. It looks like kind of like a scientific poster. It's got, the kind of intro, methods, results, population intervention, those kinds of things on it. And then there's the schematic style, which really usually details biological mechanisms. So the infographic style seems to be preferred by clinical journals. I see it more often in those. And the schematic style by basic science journals. That's not universal, but it seems to be typical. The journal that pioneered the use of graphical abstracts was Cell and the Cell Press journals, and they use this schematic style. So you see there's different approaches, and I'm going to talk about how to some different design tips for these different approaches, and we'll look at examples of both. So you could have guessed where to begin. It's look at the journal instructions and some recent issues of the journal. Some journals require visual abstracts. Others kind of leave it up to the authors. They're optional. But, I think given the potential that a visual abstract could increase the reach of your article, it is time consuming to create them, but I think it's worth it if you're trying to reach an audience that is a little bit broader. So I think it's worth doing it if you can. You should also find out from the journal instructions when you will need to submit the visual abstract. So in most cases, you only have to submit the visual abstract after the article is accepted, or maybe when you submit the revised version for final consideration. And I think that's just so, the journal isn't asking people to waste time creating visual abstracts for articles that may end up not being accepted. The other thing that you'll find in the journal instructions, and by looking through recent issues, is kind of a general sense of the aesthetic that the journal uses, and also specific requirements for the shape, the size, the layout that they use, the file format, the resolution, content, those kinds of things. And as with a written abstract, there's no point making a great one if it doesn't fit in the space that the journal gives you. And a lot of journals, you see one from the Annals of Thoracic Surgery here, provide templates, which is really nice because you can just fill in exactly what they say. So the next step is to create a sketch. And this is sort of parallel to starting to draft, creating a very, very rough draft or outline of your manuscript. And you want to think about what the single most important message that you want your readers to take away from your article is. So one place to start is to look at the written abstract's conclusion. What do you say at the very end, the message you want to leave your readers with? And a graphical abstract is even more abstract in some ways than the written abstract, in that it should focus only on that single point. If you start to try to focus on multiple things or multiple elements of the study, you will get a visual abstract that is very visually cluttered and kind of confusing. So, and definitely don't add information. This is not the place to show like additional results. This is not the supplemental figures. Um, and again, as with writing, you want to think about the audience of the journal. Are you going to try to reach other specialists in your field? In that case, maybe a little more detail or complexity might be appropriate. If you're trying to reach a broader audience, or even patients or journalists or policymakers, simplifying even more might be necessary. So you have to make decisions about what you're going to emphasize and de-emphasize based on who you're trying to talk to. And you'll want to make a few rough sketches, test out a few different concepts, a few different ideas, try different designs. A lot of people do this by hand. You can also do it by just quickly pasting things in BioRender. That's easy. Like writing, this process will be iterative. It will involve a lot of revising and refining. You may be working with a designer or artist from the journal, depending on how rich the journal is. So you want to give yourself ample time to do this. This isn't something you should probably think you're going to do like overnight. So we are editors and librarians, not designers, not artists generally. And I assume that most of you are also not graphic designers. But we are consumers of visual information. So we know more than we might think we do about what makes an image convey a clear message, or what makes the scientific figure clear. So I'm going to talk about some design principles kind of taken from basic design texts and courses, that are really easy for non-designers to use. They're things we're kind of already thinking about, but it's nice to make them explicit, and then you can sort of check and double check and apply them. The key thing I want to emphasize is keep it simple, and that pertains to everything about the figure. Keep the arrangement of the elements simple and logical. Use simple colors, fonts, arrows, text, shapes, icons, images. So you really want to think minimalist, like this painting. So the next thing you'll need to think about is sort of what the flow, how the visual flow will lead the kind of arrangement, the overall arrangement of the graphic will lead your viewer through whatever the argument is to the main point. So you want it to flow naturally. And you don't want them to have to, sit there and look at the graphic for a long time or like turn it sideways or whatever. Visual communication is intended to be quick. So to do that, you want to arrange things in an order that will feel natural and kind of like reading to your audience. So here are some common ways to do this. You can just use straight lines, either left to right, as in reading order in English, or top to bottom. That's really good for processes like time or development. You could use reading order a little differently if you have multiple lines. Use a kind of zigzag pattern where you go left to right and then left to right on the next line. Parallel boxes or frames are really nice for making comparisons between patient groups or experimental conditions. Branching structures can be appropriate if what you have is a flow chart or an analysis pipeline or a process or a method. Circles are great for things that are cycles, feedback loops. Sometimes you see them used when there's a very long linear flow that doesn't fit in the square space that the journal gives you. So you can loop it around. And then these kind of orthogonal or kind of star-shaped, cross-shaped arrangements can show multiple effects of a single factor. So I'm going to show you really quickly a few real-world examples of these. I'm going to go pretty quick for time. So but you can take a look at these on the handout and the slides later. So here's a linear flow. This is just a temporal sequence: pre-treatment, post-treatment tumor response, and then subsequent progression and therapeutic resistance. So simple left to right line. You could do it top to bottom if you wanted, but this clearly says time to a reader. Here's a zigzag style, so it moves in the reading direction. There are two lines that talk about how the study, the main points of the study, and then that big two-row span is the main finding, and having it be bigger it emphasizes that key finding. Here are two parallel frames comparing two conditions: tumor growth and tumor control. The images are very similar. They're adjacent. And it's sort of a spot the difference game. They use the paler colors to highlight what's inactivated or different between the two conditions. And you see that comes down to VISTA, which is the red molecule on the cell surface. So you can really easily spot the difference. Here's a cycle. Problem with cycles is that circles often, they don't have beginnings and ends. So if you have a cycle that goes all the way around, you want to arrange them in a way that will be visually legible or familiar to your audience. So a clock is a good model. So start at like 12 or 3 o'clock and then just move clockwise. And that people will be able to follow that pretty easily. This branching structure shows an analysis pipeline. So you have two different sets of samples, both go through high throughput assays. There are two different analyses conducted, and then those data are merged. So again, this is left to right. You could do it top to bottom. This is a methods paper. So that's an appropriate, a branching structure might work better than a linear structure for this kind of thing. And here's an orthogonal design. So this shows multiple effects of a single factor. So you have 3 possible outcomes after this immune imprinting in mice by Staph aureus. So you could have either no protection or protection, and then no protection can come from 2 different mechanisms. Most visual abstracts in reality will not use just one of those styles. It was actually really hard to find ones that did. They'll incorporate more than one. So this example was really interesting to me because it has, I think, all of those flow arrangements in it, and yet it's surprisingly legible and understandable. So it's got the zigzag, so where you read right to left and then, or left to right and then left to right again. It's got 2 linear designs that actually do different things. So this one shows the production of the cells here, or I can't even see what it says. And then here you see steps in an analysis. You have the branching structure in both of these cases. You see top to bottom comparisons, and they're actually different. So these are the outcomes of this step. And here you just have the comparison between the lymphoid and non-lymphoid tissues. So this is pretty complicated. And then you have this sort of semicircular arrangement of the relevant organs. So although there's a lot going on here, it's pretty easy to follow. And I want to use this figure as an argument for, again, planning ahead, sketching, creating multiple drafts, because I think you can imagine that the first draft of this was probably a mess and was really complicated. And the authors did a really good job of paring it down to only the very, very essential elements. So try different flows, see what works, and choose the one that is easiest to understand. Okay, second poll. What is the best color scheme for a visual abstract? So we have primary colors, neon fluorescent colors, like it's 1984, grayscale, muted soft colors, or whatever the journal tells you to use. Oh, finally, somebody picked neon. Okay, I'm gonna end the poll and show you the results a while. So it looks like almost half of you picked primary colors, red, blue, yellow, simple colors. One person went for neon, one for grayscale, about a third for the muted, less saturated colors, and whatever the journal template says. Some of those, well, we'll look at some examples of all of them. I think some of them are, say, more correct than others, or more fashionable than others, maybe. That's the way to put it. I'll come back to color in a couple of minutes, but I wanted to talk first about finally arranging the elements, the little pictures in your overall design. So once you've decided on a kind of effective overall design, you have to put the little symbols and the text and the icons and all the other stuff in there. And again, there's a few design principles that we all kind of know, but we can use explicitly. So generally, things that are similar should look similar, and things that are different should look a little different. Kind of obvious. So for example, readers will see things that are in close proximity as being related. They'll see things that are similarly shaped or similarly colored as being similar. They will see things that are arranged horizontally or vertically on the same axis as related. So here you see that the authors use these two frames with different color backgrounds to compare and contrast the neurodegeneration condition with neuroprotection with this drug, FP802, kind of linking them in that white strip in the middle. And the images are parallel, also horizontally. So we see the cell membrane and the signaling at the top, then the mouse spinal cord slices and the mouse behavior in the middle. And those two things are implied to be related because of their proximity, and they're in little boxes together. And then the organoids in vitro. So again, they're making the comparison really easy here. Another thing that they do really well here is they indicate the biological context. So you can pretty easily see that we're looking at the cell membrane, then we're looking at spinal cord slices, the mouse's behavior, and then the organoids. And they kind of show the relationship between those things with, in the vertical arrangement. I'm going to make an aside on color here, even though I'm not going to talk about color for a few more minutes. And that is that the choice of the color for the backgrounds of these two boxes is probably not random and not only aesthetic. The red often indicates, alarm or danger or something bad. And where blue is a kind of calmer color. So it's probably a deliberate choice that they used red for the background of the neurodegeneration condition. and blue for neuroprotection. Also use red for blood cells for obvious, or most blood cells for obvious reasons. Um, a couple of things that can make your figure clearer to your readers or viewers. You want to use a sans, unless the journal says otherwise, use a sans serif typeface, so a font that does not have little hats and boots. And you want to make it large enough to read. I think one of the main problems we see with figures is that, and graphical abstracts, is that folks are trying to jam a lot of text onto into a very small space, and it becomes completely unreadable. Cell suggests a minimum of 12 point. We think 10 point might be pushing it. Don't do 7.5 font. Nobody can read that. You want to not include extraneous detail. So if you have, say, a complex biological pathway, you probably don't need to necessarily, or think about whether you need to illustrate the entire thing, or think about whether you need to graphically represent every experiment you did or every analysis, and just focus on the main ones that support your main point, and focus on only the information, that the piece of the information that's relevant for understanding your conclusions. Try to minimize the amount of text that you use on the graphics. Now, this is a visual abstract, not a text abstract. You have one of those too. Some journal templates, particularly in that infographic style, tend to use more text than others. The BMJ uses quite a lot of text on theirs, but in general, keep it short. This visual abstract used very little text and is quite effective at conveying, boom, the main conclusion. It can be really tempting in various software programs to use things like word art and graphic 3D effects and fancy stuff to make the whole thing look pretty, but don't use any design elements that are purely there for decoration. Don't use, Comic Sans unless you're doing a, fourth grade science fair. These shadow effects and things like that can cause confusion in readers. Remember that readers are looking for similar things or they're looking for similarity between objects, and they're looking for information from similarity and difference in objects. So if you put that shadow in there, the purple font or something, readers may think, oh, that means something that's different from the jack that doesn't have the purple font. What does that mean? So it can be distracting and confusing and create visual clutter. So every element should contain information, not decoration. And remember also that readers very well may be looking at this visual abstract on a smartphone or on a tablet, or even on a small laptop screen. So the simpler and clearer you can make it, the better, and the more friendly it will be to your viewers. Just a quick bit on the conventions for arrows and lines. I would say people tend to jumble up their visual abstracts, particularly that schematic type, usually with like too many arrows. There are typical kind of accepted messages that different kinds of arrows send. So those fading arrows that get kind of lighter at the end and darker approaching the point of the arrow convey motion or time. Labels of whatever you need to label in the graphics should not use arrows, because arrows convey an effect or emotion. They should use the little dots at the end of the line instead. Cycles, obviously, are cycles. You can use the moving arrows or just plain arrows in cycles. A plain arrow generally is like, this affects something, it was done to something, it's just moving, it just activates moving forward. You can indicate the strength or weakness of an effect by making the line and the arrow thicker or thinner, or use a dotted line for weaker or weakened effects. The tapered arrow, which is a little bit different from that fade arrow, can indicate the direction of change. So if something's getting greater, you can make the arrow get bigger, or if something's getting smaller, you can make the arrow get smaller. Inhibition is usually indicated with the T at the end of the line. And you can also use color and arrows to indicate things like upregulation and downregulation. Color and or arrows, whichever you like. And just be aware, if you can, at all costs, avoid crossing lines and arrows because it just gets really visually pretty messy. Color is really important in conveying information. It can also look pretty, but it should highlight your main points. So in this image of this macrophage, we'll start with the one on the left. The nucleus is kind of bright purple. The bacteria are bright orange. The phagosomes in there look like they're sort of the same color as the bacteria. There's these other bright pink granules in there. So it's not particularly easy to see what the author wants you to be looking at. In the second one, they've desaturated the colors to use shades of blue for everything inside the cell. And then this highly contrasting color for the bacteria. So you can get the message there much more easily that what we're looking at is phagocytosis and it's of these bacteria. So the contrast in color there really creates a much clearer, simpler message. And one way to check this is to see if you're doing this effectively is to convert the image to grayscale and see if the contrast is still apparent. BioRender actually just has like a button that says flip to grayscale and you can see it and then it flips it back. Again, consult your journal template when it comes to color. Some journals use this like super bright, eye-popping color, really attention grabbing. So, if that's what they use, then that's what they use and ignore this. And my last piece of advice is to keep your abstract or your visual abstract concise, just like your written abstract. So these examples are from the Cell Press guidelines, and the before abstract is what the authors submitted, and then the after is what the revised version that Cell's graphic designers fixed or revised, let's say. So you can see some of the things they did here. They condensed that 3-panel rectangular design into 1 panel with 2 components according to journal guidelines. You can see that this panel on Hira has been taken out. So they just deleted this part because it's not really relevant to the main point of the paper, which was this comparison of wild type and ATRX null conditions. So before, the relationship between like, what does that have to do with that, wasn't clear. There's a lot of redundancy in these figures. So you see like almost the same networks and there's like X's and it's just really hard to figure out like, what are we supposed to be looking at? And this 2-panel organization with all the common factors together in the middle makes the whole thing just a lot clearer. They removed this like teeny, I don't know if you can see my pointer, there it is. But they removed this teeny tiny, which was 6.5 point font, and some of this extraneous labeling of the text, and got that out. And then again, moved some of this repeated, these repeated graphs. And the other thing I noticed they did was they added they added some color and they added particularly red to indicate the crucial point, which is this upregulation of TERA. And then last, I want to just refer you to some resources. These are all also in your handout that will be coming. BioRender is a really pretty powerful web-based tool for designing all kinds of visual graphics, icons, templates, graphs specifically for biology. MD Anderson has an institutional license, which Larissa can tell you a little more about how that works. And that's a premium account, so you get all the features. The Noun Project is a great place to look for those little infographic style graphics on anything you can think of, from abstract concepts to concrete ones. Their free plan has some limited features. If you or your lab or your PI wants to pay for the paid subscription, that gives you access to the full features. The institution doesn't have access to that. And MD Anderson Strategic Communications Office has medical illustrators and graphic designers in it who can help you with some of the overall layout. If you need illustrations of a particular, anatomy or surgery or something like that, they're able to help you out with that. So with that, I'm happy to answer any questions you might have. Ran a little over as usual.
We did. There were a couple of comments in the chat.
Oh,
It looks like
let me look at the
someone
chat.
pointed out that when talking about color, keep in mind that some viewers may be colorblind.
Yes.
I noticed all the examples, there were red and blue were the typical
Yes.
contrasting colors. So I think
Yeah.
there's a little bit more awareness of that maybe
Yes.
than there was in the past, but that's a good thing to keep in mind.
We had a previous Dear Editor that Erica and I did on figures, and that has quite a bit of that is about making your figures accessible to colorblind readers. Yeah, try, I mean, in just quick terms, try as hard as you can in all your figures not to use red and green to represent salient contrast, because a good proportion of your readers might not be able to see it.
The other question I see here is how do you think about abstracts, or sorry, think about graphical abstracts in terms of reviewing because they're so comprehensive? Do you send
Yeah.
them before or do you send them after your article has been reviewed by a journal? Which is a
Oh,
great
I thought,
question.
did you mean before or after it's been reviewed, or in case of review articles, like a narrative review that's just so
Case of reviews, maybe
comprehensive?
that too.
Could you clarify what you meant? In the case of like when it goes okay.
Both.
Both. Okay. In the case of when it goes in terms of the review process, I would say most journals that use them ask for them after the initial peer review. So obviously they're, if they're going to desk review or desk reject something, they don't want you to have spent the time making the graphical abstract. And they know that, if you end up submitting it somewhere else, you're just going to have to revise it. They kind of understand that that's a lot to ask. So what they so normally I see them required either at the stage where you're revising and it looks like they're probably going to accept it with revisions, or even after the at the acceptance stage where they're going to start doing the copy editing and the formatting and things like that. So normally you don't see the visual abstract required at initial submission. In terms of review articles, gosh, I just worked on one of these, and it is really challenging when you have a lot of information, as in a narrative review with 200 citations that's 30 pages long. It's really hard to get it all in one place. But I think, even a narrative review article should have a conclusion. It should have some sort of important message. And so I would say focus on that rather than all the details of all the pathways or all the drugs or all the things, all the different surgical techniques that you're reviewing, and instead focus on what your kind of main message is. Or you could even maybe divide it into blocks for each, for different sections of, parallel to the sections of the manuscript that deal with different questions, something like that. Sort of another, just, look at some review articles in that journal and see how they did it. I see, when I was doing this, I saw a lot of journals require graphical abstracts, or they use them for research articles and not for review articles, although I know some review articles, some journals do use them for review articles. Yes, I don't know what communications does. I know that communications is available if you contact them usually well ahead of time. They can consult with you on and help you design an effective figure. I have worked on a couple of those where you send the author sent a literal sketch on a post-it note, and they kind of tried to put it, they put it in a more a nicer looking format and made some suggestions about how that could go. We, as the editors, are happy to, I mean, again, we're not designers like communications is, but we're happy to look and make sure that it looks clear. We do read things from the point of view of, a non-scientist an informed non-scientist who would be reading your article. And we can tell you if everything is legible and logical and flows nicely and really and matches the manuscript's conclusions. So I think together with communications, we have a comprehensive ability to help you out.
Yes.
Does anyone, Erica or Larissa, do you know if communications charges? We don't. Our services are free.
No, we don't. I'm not sure if
I
Michael.
think they stopped charging for them.
Okay, good.
Okay.
Not that long ago.
Okay, good.
Okay.
And if anyone wants to try out BioRender, we're going to send out the handout. You can just log in and try it. We do kick people out after 90 days of inactivity, but all your materials stay in there. And that's just because we only have so many seats. But you can try that out and see if it works for you.
Yeah, and if you find that your account is, if you haven't used it and it tells you that you've been kicked out, you just can log in and all your stuff's there.
Yes. Larissa put into the chat you can always email the library and we can answer questions there generally. If you're watching this presentation today and have questions afterwards that we didn't answer or you didn't think about until after, you can always email us. You can also post on the team if you like, which, as I said earlier, I will post a copy of these slides and additional list of resources that go along with this talk, as well as a link to the recording if you missed it or if you missed part of it, so you can go back and look at it later.
Or if I talked
but if you
so
have
fast that you
questions,
didn't.
yeah, but if you have questions about this or anything else that we talk about in Dear Editor, you can always feel free to ask on the teams. And we'll take a look and do our best to answer it or point you in the right direction anyway. Not seeing any other questions. Larissa did put a link to BioRender in the text in the chat for those interested. Since we are past, I think I will go ahead and wrap up. And again, if you have questions, always feel free to contact us. And thank you so much for joining us this afternoon.
Thanks, everyone. Good luck with your graphical abstracts.